So in the last video, we have discussed about the potency of the stem cells, and depending on that potency of stem cells, we actually can classify them into different groups. So we have talked about the totipotent and pluripotent stem cells. Here are a few notes or few information that you need to know about this. One is the pluripotent stem cell can be isolated, adopted, and propagated indefinitely in an undifferentiated state as embryonic stem cell. So this embryonic stem cell, what I have said, this embryonic stem cell we can collect from the embryo. We can collect cells from the embryo, and these are stem cells. As you know, in the early stage, these cells have enormous potential to differentiate into all the lineages that a cell actually that a body actually possesses. So you can use this embryonic stem cell as a pluripotent stem cell. You can culture them. This is very important in research research related to the stem cell. And totipotency, how much they actually stays in the developmental process, this totipotency in stem cells is actually only up to 4 cell stage, sometimes 8 cell stage of the embryo. The zygote is the 1 cell stage. You know, the zygote starts divide, they will produce two, and once they produce 2 cells, if one more division, that will produce 4 cells. So till this fourth stage or the next stage for the 8 cell stage, these 4 or 8 cell stage from 1 to 8, this is actually the totipotent stage of the stem cells. All these cells are totipotent in nature. There are a few controversy that whether we should call the zygote as a totipotent or not. We can actually know about it in later videos. And another thing is, experimentally totipotency can be demonstrated by the isolation of a single blastomere from pre-implantation embryo. Yes, when they are not there is no implantation of the embryo within the embryonic wall or the uterus wall of the mother, you can collect them, and the totipotency you can as they in totipotency. What is important, that to produce the extraembryonic membrane is very important. Once the cells or the structure has loosened the capacity to produce the external membrane, that is to form an association, or that is very necessary for the implantation of the embryo in the mother's body. Once it has lose it, so it will become the pluripotent. Before that, it is actually totipotent, right? Because it has the capacity, these cells has the capacity to produce the cells that are necessary to produce the extraembryonic membrane. Okay, so these are very few informations that you need to keep in mind. So one thing is that I have talked about the differentiation thing very largely. I have used this term very largely, right? So in this case, what we need to know, how this differentiation actually occurs, or this differentiation or this specialization, how it actually occurs, we need to know. So this is very crucial. You have to have the idea of the genes. I know you all have the idea of gene, and we know that with the presence of DNA, with the presence of active DNA of the gene, they are producing few important proteins that are necessary to maintain our body in the form as we want to, as our body want to remain in, or the cells actually are wanting to remain in. But what happens when this is completely in a genetic level? If any transcription factor or any important signal from outside actually comes and tells a specific gene to produce a specific type of protein, and when this protein is actually produced, it is telling the cell to function in a specific way. So a specific signal via the production of the protein, you know about the signal transduction process. There are different signaling processes are important in our body. So the signaling processes via the outside different proteins or different outsides out signal from outside, they are telling the cell, telling the genes to produce a specific protein, and with in response to that protein, that cell is behaving like that. And at the time this behavior is becoming a changed one, when this behavior is actually changed, the cell is becoming, for say in some cases, they're becoming differentiated. So these specific proteins which are important to tell the cell that you need to now go to another pathway, you need to go to another lineages. So this signal is coming, and with these signals this cell actually are becoming specialized in nature. There are different ways to do this in cells, different, like I've just told you about the transcription factors. They are controlling the genes. I have told about the potency. So there are very important few specific transcription factors that are actually regulating that which potency one cell will remain. They are controlling this thing. They are controlling the genes. When they are activating the gene, they are actually producing in that certain way. We will talk about the BMP4, OCT4, noggin, all other octave, different types of transcription factors is actually regulating these transcription processes. Okay, so specialization of cell can be done via different processes. One is like there is uneven distribution of transcription factors in the oocyte. When oocyte, the zygote actually forms via the interaction between the oocyte and the sperm. What happens? Sperm actually releases the genetic material that comes within the oocyte body, and they fuses. And when this zygote is formed, they have no time. They start to the process of dividing. And when they actually you can say that this is the in the left side the picture, this is a oocyte. Okay, and the few transcription factors were in the lower side of this oocyte, in the lower cytoplasm of this oocyte. But the cell actually, you can see this red structure, red circle I have, half circle I have drawn here. So the division is when this division is happening, and it is happening from the middle of this cell. One part of this oocyte have the maximum amount of transcription factor that may produce another type of cell, and the upper side is lacking this transcription factor. So this is taking no time, and this division is actually happening. So with time what happens, the region, the cell, this oocyte will produce two cells, and the cell that will have the more number of transcription factor of a specific signal for say that cell, that after division that cell will produce this type of, they are prone to become that type of cell. They are prone to go into that lineage. But the cell which will produce from the upper side or the other side of the cell oocyte, that may not have those signals, those transcription factors to go into that lineage. So this is one important thing, how the cell actually specialized at the beginning, when there is a very initial developmental events are taking place. Second one process is signal from adjacent cells. So if two cells are actually residing one after other, so there are few junctions, as you know, there are few junction, gap junction, tight junction, all these junction actually, with these junctions these cells actually can communicate with each other. So if a cell in my side, two cells are residing beside one other. So one cell, via this junction, tight junction, gap junction, all these junction, they are sending few signal to the adjacent cell, and after getting these signals this cell is actually behaving like that. The localization, tissue localization, where one you can see in a tissue the cells that are residing in a colony or in a place, they are actually similar in function and similar in structure. How this is actually happening? They are actually, when this developmental process is going, and at a later stage of this developmental process, via these adjacent cellular signals that are coming from the adjacent cell, via this junction that is telling the cell to behave in that way. And other is the external stimuli, when the stimuli is actually coming from the external cells. So this external any other proteins coming from the other side of the body, it is coming here, like the hormone function. Hormone function, they are actually originated from one region of the body, from endocrine gland. They then travel via the blood and go to the other part of the body, and they function there. In that case also, in here also, in external stimuli, what happens is stimuli actually come from the other side of the body. There are few receptors that are present in the cell membrane. Cell membranes actually capture those those proteins, and they get a signal. They then send this signal via the cytoplasm. All these proteins, carrier proteins, they come into the nucleus. They will regulate the gene, and this body will then function accordingly. So the signals are sometimes contact dependent when there is a contact between two cells, and sometimes, like I have said, they are contact independent when they are not producing any contact between uh, themselves. So this was about the differentiation, how differentiation actually is helping the different formation of different type of body cells within our body. And the stem cells, I have talked much about the differentiation. Like all these signals is actually regulating the stem cells to become the differentiated one. But how these stem cells, what are the genetic what are the regulation in the genetic level, we'll know about that in the